So at first, let me use the term. At first, just today we have today I have taken up the a new topic of the genetics, which is the extra nuclear inheritance, or that is cytoplasmic inheritance. It is also known as cytoplasmic inheritance, extra nuclear inheritance, maternal effect, or uniparental inheritance. You all of you know that the genes are present not only in the nucleus but outside the nucleus. The genes are also present in the cytoplasm and cytoplasmic organelles, and the two very important cytoplasmic organelle, which include the mitochondria and chloroplast, which have their own genetic material consisting of double-stranded circular DNA. And such DNA are known as mitochondrial DNA or chloroplast DNA, respectively, in case of mitochondria and chloroplast. And you know that the genes are of, just I have said to you that the genes are of two types, nuclear gene and extranuclear gene. The genes which are present on the chromosome inside the nucleus are known as the nuclear genes, and the genes which are present outside the nucleus and inside the cytoplasmic organelles are called extranuclear genes. And the branch of genetics which deals with the extranuclear genetic system is known as organeller genetics, because the genes are present in the cell organelle. So this branch of genetics which deals with the genes present in the mitochondria and the chloroplast constitute the organeller genetics. You know that most of the characters are controlled by the nuclear genes, but there are various examples. There are various characters or traits which are controlled by the organeller genes, like the mitochondrial and the chloroplast genes. And you know that the term genome is used. The term genome is used for the genes present inside the nucleus, and the total number of genes present on the haploid set of chromosome constitute genome. And the word plasmon is used for the genes present in the mitochondria and the chloroplast, and especially the genes present on the DNA or DNA of the mitochondria is known as chondriome. Total number of genes present on the mitochondrial DNA constitute the term chondriome, and the total number of genes present on the circular double-stranded DNA of the chloroplast constitute the term plastome. And collectively plastome and chondriome constitute the word plasmon. So I think that that you are quite aware with the term genome and plasmon. And plasmons are of two types, chondriome and plastome. Now look at now there are various examples. Let me at first point some important features of the uniparental inheritance or cytoplasmic inheritance. In case of extranuclear inheritance, which is also known as cytoplasmic inheritance, which is also known as uniparental inheritance, cytoplasmic inheritance, extrachromosomal inheritance, or maternal effect. In this case, the characters or traits are controlled by the genes present either in the chloroplast or mitochondria. Maybe the very important point that how does the what is the difference between the inheritance, nuclear inheritance and cytoplasmic inheritance? If you make the very important point you have to know is that if a reciprocal crossage do not give identical result in case of extranuclear inheritance, whereas the reciprocal crossage give identical result in case of nuclear inheritance, as you have seen in case of Mendelism. If the genes are controlled by the nuclear genes, and if you make a reciprocal crossage, you will get the identical result. But if the characters concerned are controlled by the mitochondrial and plastic DNA, you are not going to get the identical result if you make the reciprocal crossage. Okay? Look, there are various examples of the cytoplasmic inheritance. I have just at first I would like to provide you and explain this cytoplasmic inheritance with a very common example of the plastid inheritance. Plastid inheritance in case of Mirabilis jalapa, which is a 4 o'clock plant. Look at the whiteboard. I have shown the crossage made. You know, in case of you know that in case of Mirabilis jalapa, there are three types of branches. Three types of branches. Some branches, so there are three types of branches with respect to their color. Branches may be kya? Branches may be white, branches may be green, branches may be variegated. And you know that Mirabilis jalapa produce the kya? Bisexual flower. So in this case, three different cases, three different types of cross have been made, which I have shown on the whiteboard. Now look at the whiteboard. Look at the whiteboard here. Just I have written here the things which I have already mentioned is also written on the whiteboard. Nuclear genes. Look at nuclear genes. Genes are of two types: nuclear genes, extranuclear genes. The term genome is used in case of nuclear genes. The term plasmon is used in case of extranuclear genes. These plasmons are of two types. A plastome is the DNA of the genome present in the chloroplast is known as plastome, and the genome present in the mitochondria is known as chondriome. Now look at the one example of the cytoplasmic inheritance, plastid inheritance. Look here, I have written here plastid inheritance in Mirabilis jalapa. There are three types of branches. Green branches will have the chloroplast, white branches will have leucoplast, and the variegated branch will have green chloroplast and leucoplast. This is the mixture of the green and white. If a cross, look here, I have shown here three types of crosses made. In first case, in case 1, case 2, and case 3. In case first, the female branch, a cross has been made between the a cross has been made between the male and female, and the phenotype of the female branch is the green, and the male branch maybe, and the pollen may be taken from the green branch or white branch or variegated branch. This is very just I want to clarify it here, that if you pollinate the flower present on the green branch, okay, if you take the pollen grain from either from the green branch or white branch or variegated branch, and if you pollinate the pollen either coming from the green, white, or variegated, and the female flower present on the green branches, what will happen in F1 generation? All the branches will be green. In F1 generation, all the branches will be green if you use the green branch as the female source, and the male branch either as a green, white, or variegated. The main thing is that you know that it is the egg which will contribute the chloroplast or chloroplast and leucoplast, or both of them will come from kahan se? Just I have told you that the cytoplasm is transmitted transferred through the egg. If you use the female branch, if you use the green branch as the female source, and if you take the pollen grain from either from the green branch or from the white branch or from the variegated branch, what will happen? Because the egg is present on the white, egg is present on the green branch. Therefore, in F1 generation, all the branches will be green, because the plastid is being transferred to the zygote through the egg, and egg is present on the green branch. Therefore, all the branches in the F1 generation will be green. Okay? In case 2, look at the case 2. In case 2, the female source is the white branch, and the male source is either green or white or variegated. It doesn't matter whether you are taking the pollen grain from the green branch or white branch or variegated branch. Your flower is located, egg is located on the white branch. Therefore, all the branches in the F1 generation will be the white branches, because the leucoplast is being transferred to the zygote. Chloroplast is not being transferred to the zygote. Therefore, all the branches will be white in F1 generation in this case. Let us look at the third case. In case third, mein, the female source is the variegated branch. What is the meaning of variegated branch? That the egg will be having both the chloroplast and leucoplast. and the male source and the male source may be green, white, or variegated. What will happen in the F1 generation? In this case, the branches may be green, branches may be white, or branches may be variegated. So this the very important point you have to learn here, that it does not matter whether the male gamete is coming from the green, white, or variegated. Whether the male gamete is in all the three cases. Look here, in all the three cases, the male source may be green, white, or variegated. Male source may be green, white, and variegated. Male source may be green, white, and variegated. So the main point here is that it doesn't matter whether the male source, whether the male gamete is coming from the green branch or white branch or variegated branch. The point which will matter is that whether the female source is from the green branch or the white branch or the variegated branch, and the phenotype of the progeny will depend upon the phenotype of the female branch, because you know that the female branch will provide the egg, and you know that it is the egg which contributes cytoplasm to the zygote. Therefore, if you use the green branch if you use green as a female branch, all the branches in F1 will be green. If you use the white branch as a female source, all the branches in the F1 generation will be white. And if you use the variegated branch as a female source, all the plant will be having green, either green or white or variegated plant. This example shows that it is the cytoplasm which contributes it is the female source, it is the egg which contributes the cytoplasm to the zygote. And in this case, it is the cytoplasm which contributes plastid to the zygote, not the male gamete. And the plastid may be green, may be white, or maybe a mixture of green and white. And the same thing has been shown in this diagram, in this structure. Look here, the central circle. There are three rings: the first ring, the central ring, the middle ring, and the outer ring. The inner ring, the middle ring, and the outer ring. The inner ring, the central ring, represents the female source. The middle ring indicates the male source, and the outer ring and the outer circle shows the progeny. What will happen? Look here. This is the best method to explain the things. This matter, this topic. Look here. This is the central ring has been divided into three parts. This is shown if this is the green branch. Just I have shown three process. The same process has been represented by this diagram. If you use the female source, if you use the female source as a green branch, and the male source either green or white or variegated, okay? All the progeny will be green, depending, because the female source is green. All the progeny will be green, whether the male is green, white, or variegated. Similarly, if the female source is having the leucoplast, the male may be variegated, may be white or green. All the branches in the F1 generation will be